drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. If you like to read like I do, then you should check out the ReadWise app. It's totally free to download, and if you go through the link in the episode description or on my support page, you can get a free 60-day trial for zero cost. ReadWise is a really cool app that allows you to import digital highlights from over 30 different platforms such as Kindle, iBooks, PDFs, and more. It makes it really easy for you to review and find and share excerpts and quotes from whatever it is that you're reading, whether it's PDFs of scientific papers or books that you're reading on your Kindle, and you can even save and use highlights from physical books. I read a lot of physical books before bed, for example, or when I'm traveling, and the ReadWise app actually integrates with the camera on your phone so that you can take a photo of a page that you're reading, highlight a specific chunk of text from that page, and export it to the app so that you can reference it or find it or share it later. They even allow you to export really cool looking graphics of some of the highlights that you have in the books or the papers you're reading so that you can share them in an engaging way. So check out the ReadWise app today if you are a reader and you like to reference and organize what you read. Go through the link in the episode description or on my support page or in my free weekly newsletter at mindandmatter.substack.com and try the ReadWise app 60 days free today.
Yeah, it's good to be here. Good to meet you.
Do you want to just briefly start off by telling everyone a little bit about who you are and what you study?
Yeah, I'm Mike Levin. My background is first computer science and then cell and developmental biology. I run the Allen Discovery Center at Tufts University, and my lab works on many different issues around how cognition manifests itself at all different scales and at all different kinds of embodiments.
Mm-hmm, mm-hmm. And so cognition can manifest at different scales, meaning not just the organismal or whole brain level. We think it might manifest at smaller scales.
Correct. What we do is we take approaches, both conceptual and practical bench tools from neuroscience, and we apply them to all kinds of things that are evolved, engineered, and hybrids. So this means cells. This means trying to study memories in cells and tissues. It means trying to communicate goals to organs in biomedical settings. It means looking at materials and hybrids and cyborgs and various things in terms of what kind of minds do they have, what kind of processing can they do, and how many of the tools that we have in behavioral science can be applied far outside the brain.
Mm-hmm. And so let's start off by just having you talk about, like, what is cognition? Do you have a good definition that is applicable at all of these different scales? How would you define that as a way to get us thinking about what it is? And then we'll probably get into information coding and the different ways that might manifest in different biological systems.
Yeah, I can give a rough definition. I don't get too caught up in definitions per se, because I think especially as we apply some of our concepts outside their normal areas of application, the definitions can often hold you back more than they facilitate. And so much like with defining life or those kinds of things, I don't think that sharp definitions are doing us that much good. I tend to operate from a very kind of pragmatic operationalist perspective. So in other words, I don't want to argue about the definition of cognition, but I do want to point out that isn't it interesting that we can take tools that we've been using to study brainy behavior for many decades. We can take those tools, and I'm talking about everything from optogenetics, neurotransmitters, perceptual multi-stability, active inference, just across the spectrum of practical and conceptual frameworks that we study in brains. And we can show that those tools allow us to discover new science when you port them elsewhere. Mm-hmm. So my point is simply this: if the tools can't tell the difference, then you're not able to say that, well, that's not real cognition. That's sort of as if cognition. It's real if the tools are helping you find new science. That's how I see it. So by cognition, I mean a very wide spectrum of properties, all the way functional properties, all the way from simple learning, habituation, sensitization, associative conditioning, predictions, being able to make predictions, being able to hold counterfactuals, planning, all of those things. We haven't claimed, for example, language use in any of these things. So I don't, obviously language is part of cognition. We've never seen an example of proper grammar, at least yet, in these things. So I don't say language, but most everything else that I've just mentioned exists far outside the brain.
Mm-hmm, mm-hmm. And so it's usually pretty intuitive for people to think about information and cognition when it comes to the brain and the nervous system, because that's the organ most identified with cognition. And then, you learn about action potentials and spiking, and okay, this makes a lot of sense. These cells have a behavior that gives you a code, right? They can spike with a certain pattern, and they have this all or nothing information code that is, it's very intuitive to think about how this can serve as a binary coding scheme that can, produce cognition and behavior and all the stuff that brains do. But there's also examples out there, I think, of analog coding in biological systems. And that's not something you usually hear about as often unless you're sort of deep in this world. What is the difference between analog and digital coding? And what are some examples to get people thinking about analog systems in biology?
Yeah, let me take one step back before we get into that, just to kind of set the stage a little bit. If we ask ourselves so what I think the neuroscience is not the study of neurons. I think neuroscience is the study of the cognitive glue that binds competent subunits into higher level cognitive agents. So you and I are more than the sum of a bunch of neurons, because there's a set of policies and mechanisms that allow up a very particular kind of networking to form. And so that's electrophysiology that allows our neurons to work together, to have memories, goals, preferences, and to have computations that individual neurons are not doing. That's not to say they're not doing any, they're doing some, but they're not the same as what you and I are doing. And so we can ask then that question, okay, so that's a remarkable, that bioelectricity as or electrophysiology as a kind of cognitive glue in that sense, where did that come from evolutionarily? Like, how old is it? Mm-hmm. And it turns out that evolution discovered that electrical networks are really good for this kind of thing. So integrating or synthesizing information across space and time, storing memories, doing computations, that was discovered by evolution around the time of bacterial biofilms. It's extremely ancient. So all of the same components that we study in the brain, ion channels, electrical synapses or gap junctions, neurotransmitter machinery, all of that stuff is incredibly ancient. It predates multicellularity, and it's homologous. The kinds of things I'm talking about are evolutionarily homologous with the things that people study in the brain. And so the next question you would ask is, okay, so if these things were around before brain and muscle were here to move us in three-dimensional space, which is why the kind of digital coding is so prevalent, what were these networks thinking about before? And my claim is simply this: that everything we do in the brain is an evolutionary pivot of using these exact same mechanisms to traverse a different space. This is long before nerve and muscle came on the scene. That is a space of anatomical shapes. So here's where the analog computation is going to come in, because you have a bunch of cells. The cells form a collective intelligence, very much like we are a collective intelligence made of neurons. But cells were assembling into collective intelligences long before there were neurons or muscle. And what that set of electrical networks was doing is thinking about how to move you from where you are at the beginning in the anatomical space, you're one cell, you're a fertilized egg, how to move you to wherever you're going to be. And it's not a simple, it's very much a navigational task that requires all kinds of protocognitive competencies. It is not a simple hardwired, well, follow these steps and here you go. And I can discuss why that is. But it's very much a navigational problem solving process. It's actually what I call an intelligent process because it has the ability to solve novel problems. And so that process uses analog computation because the way that those electrical signals are interpreted are not the way that neurons in the brain are interpreting their activity using this digital coding. Because what a couple of things happen in pivoting from developmental to behavioral controls is that, first of all, the speed had to go up immensely. So we're talking about processes that take hours, sometimes days, as opposed to milliseconds. So the speed had to go up, and then there's some other benefits.
You're saying that when an animal is developed and formed, we have second by second behavior and things are happening at the millisecond scale in the nervous system to govern our behavior. But when you think about something like development, the timescales with which these things are happening are different, because it can take hours or days for something to play out when you're thinking about something like the formation of an organ or a limb or whatever.
Correct. And what's playing out are decision-making, sensing, perception, decision-making, attention, recognition, all of those same things, but they're stretched out in time. And, we're not very good at visualizing behavior that happens at very slow or very fast timescales. And so this is why we have separate departments of developmental biology and neuroscience. But we made a tool. I used to have my students do this by hand, and now there's an AI tool online that will do it for you. You can take pretty much any abstract of a neuroscience paper or a behavioral science paper, and all you do is you put it in a word processor, and you just do a find replace. And everywhere it says neuron, you just say cell. Everywhere it says millisecond, you say hour. And you could make a couple of other tweaks, but basically that's it. You have yourself a developmental biology paper. I mean, it's amazing how much symmetry there is between these two fields. And I think it's no accident, because evolution is telling us that this is what happened. So that's where you have an analog code, because the interpretation of the bioelectric states that go on in the body as it's trying to figure out how many fingers you're supposed to have and all this kind of stuff. Those patterns they're analog. They're spatial distributions. Sometimes they actually look like the thing that they encode. Sometimes they look nothing like the thing they encode, and it's a lot of hard work to decode it. Much like neural decoding, people are trying to do that in the brain. And we pay a lot of attention to how the interpretation works. And it has a little bit of an analog flavor of a digital flavor, in the sense that there are ways to using this bioelectric code, we're trying to crack it exactly as neuroscience is trying to crack the code in the brain. And some of the messages in that code refer to specific organs. So we know a message that says build an eye here, and we can reproduce that message elsewhere, and you can get eyes in the weird parts of the body and so on. So there is a kind of a digital aspect to it a little bit, but the patterns themselves fundamentally are very analog, and they're very fluid.
And let's just give people a concrete sense for how analog information can live in biological networks. So you mentioned things like gap junctions earlier. Can you just talk a little bit about analog signaling between cells and what that looks like in contrast to something like the spiking or action potentials that neurons are associated with?
Yeah. There's the one thing that is basically so let me list the things that are exactly the same, and then the things that are different. So things that are exactly the same are that pretty much every cell in your body uses ion channels to set up a resting potential. So sodium, chloride, potassium, occasionally protons.
Yeah, but not just neurons. This is just basically all cells.
Every cell in your body is doing this. Okay, they're using ion channels, ion pumps, ion transporters, co-transporters, all that stuff is being used to set up a particular voltage gradient between the inside and the outside of the cell. So the resting membrane potential. Now there are gap junctions, which are electrical synapses, which sit in the membrane of cells. There are 6 on one side, 6 on the other side. They dock like two submarines trying to dock, and they allow small molecules, in particular, like very small molecules, so in particular charge, to go across, but also some other things that are not particularly ions as well. And so that allows you to set up an electrical network of cells that just like in the brain. So that part's the same. Some of the things that go through these gap junctions are neurotransmitters, things like serotonin. So that part's the same. The network itself is processing information that then it gets read out by other cells, and other actions take place based on what the network is doing. That's just like neuroscience. Here are some things that are different. First thing that's different is that neurons mostly like to sit at a particular highly polarized resting potential, and then when they get activated, they depolarize and they try to get back there as quickly as they can. The cardiac cells do it too. Most of the rest of the cells of the body are much more much more flexible at what voltages they sit at, and B, those voltages change slowly. So they don't spike. They tend not to spike on a millisecond scale. They tend to change slowly as a function of computations that are happening in the network. Mm-hmm. So they may change in minutes, hours. We make videos of these things. We use all the same tools. So we developed the first use of voltage sensitive fluorescent dyes to track all these things. And so we make videos of what this actually looks like. And of course you have to time lapse it and then speed it up, because otherwise, it takes too long. But you can see all the electrical conversations. You can see waves, and you can see borders and boundaries. And here's where this pattern we call the electric face that Danny Adams in my lab found years ago, which literally tells you where all the organs in the face are going to be. Here are the two eye spots. Here's the mouth spot. Here are the placodes on the side. Mm-hmm. So, and all of those changes are slow and they're gradual, and the voltages can be anything. So a cell might be -30, and it might be -42, and it might be, and the other important thing is, much like in neuroscience, is that it's generally not the voltage of any one cell that determines what it's going to do. This is not a single cell level code. It is the spatial pattern across a whole bunch of cells that determine what everybody's going to do. It's very much an emergent network property, and it has a virtual governor kind of aspect, just like the brain does.
Mm-hmm. So no one cell's in charge. You've got a bunch of cells. We could be talking about a developing organ here. We're not talking about neurons. We're talking about some other types of cells. They have a membrane potential of some kind. They are electrically coupled. They're plugged into each other, literally, and ions can flow between them. And so these membrane potentials can change slowly in an analog manner, but you see similar phenomenon as you would see in the nervous system. You see waves of activity, but there are differences. One of them is time scale. Things are basically playing out more slowly in, say, a developing organ than they are in what's happening in our brains right now to move our hands and mouths and things like that.
Yeah, and the other thing to keep in mind is that when I say the gap junctions allow current to equalize between two cells, this is not just a giant syncytium where everything just ends up at the same voltage, because both many of the ion channels and the gap junctions themselves are voltage sensitive. So what you have there is a voltage sensitive current conductor, aka a transistor. So all of these things mediate very rich feedback loops, both positive and negative feedback loops, and that's why it can self-regionalize. So it's not. I like to think it's not an accident that Alan Turing, who was very interested in intelligence and computation and other media and reprogrammability and all of that, he also wrote one of the first papers on a chemical model of morphogenesis, trying to understand how does order come spontaneously from a well mixed set of chemicals. Mm-hmm. And I think I think he was onto this idea that there's a profound symmetry between the self-assembly of bodies and the self-assembly of minds, and that evolution happens to like bioelectricity for all of these things, but there's a much more fundamental thing going on here, which is that you have to align and coordinate your subunits towards higher order goals in spaces that they know nothing about. Mm-hmm. And this is, I think, works the same way.
Yeah, and when we start to think about some of this stuff, it gets interesting and I think difficult. So when you have a higher order goal, I guess the question I want to get to is something like, if we talk about morphogenesis and some of the things from the developmental biology side of this, how does a clump of tissue that doesn't have neurons, doesn't have a thing that we normally think of as the thinking stuff, how does it know, if I want to make my hand and I've got 5 digits, and it's supposed to have 5 digits because most of us have 5 digits. Occasionally that goes wrong and some people might have more or less, but I'm supposed to have 5 digits in this morphological pattern in this exact shape. How does the tissue that's developing into my hand have a goal in mind, so to speak? How is that actually encoded in terms of these voltage maps that where these analog information signals live and propagate.
Yeah, I mean the first thing that just to mention, and people say this all the time, is how could a clump of cells have goals, especially if they're not neural cells? And they say, well, how do your neural cells have goals? And then, yeah, I guess I don't know. Although at some point, that's not a great answer to this. Although at some point, say, well, it has something to do with the bioelectricity, the electrophysiology. I say, perfect, exactly the same way. And so, also to be clear, when I say goal, I don't mean some sort of human level metacognitive. I know what my goal is. I'm happy about my goal. I don't mean any of that.
Yeah, but there is a destination that's pre-specified.
Absolutely, and so I just want to point this out, that I'm not claiming second order metacognition. What I'm saying is, as in a basic a homeostatic cybernetic type of system, there's a recorded set point which serves as the target of an error minimization scheme. So that what's happening here is that there is a set point, and you've got a system that's able to measure current state, it's able to access the memory of the target state, and as long as those two things are far enough apart, it's going to work hard to try to reduce the error to acceptable levels, right? So this is, we have, it's no longer magical to say that machines have goals. We now have a mature science, and cybernetics and so on, where we can actually cash this out in mechanisms. And so this is of course not how people describe developmental biology normally. What developmental biology is, typically the story is described in a very open loop, feed forward kind of way, that you have a bunch of cells, they follow local rules, almost like a cellular automaton, they all follow local rules, and then they say, well, then there's emergence, meaning that if you do enough things in large numbers, eventually something complex will emerge. So that is so I'm going to say here that is true. Emergence of complexity certainly does come from rules, but that is a very small part of the story, because what we see, and so now I'm going to tell you why I think they have goals and how those goals are encoded. Because what you have here, typically, with the exception of certain things, maybe C. elegans, the nematode might work like that, maybe, but most things don't work like that. In embryonic development, in regeneration, in cancer suppression, you have a very active, continuous monitoring of large-scale properties and a repair to those properties if you introduce an error. And I'm just going to give you two examples. And by the way, I treat development and regeneration as exactly the same, because I think development is just drastic regeneration from one cell. It's still an error minimization scheme, and it uses all the same components. Like development is just regeneration after you've been pruned down to one cell. But so the one that most people would have heard of is axolotl limbs. Yeah. So you got this amphibian, and you chop off the limb, or they bite each other's legs off, and the cells very quickly start to proliferate. They grow, they make a new limb, and then they stop. That's the most amazing part of this whole process. It's not the growth, it's the stoppage. Because when do they stop? They stop when a perfect axolotl limb has formed. That's when they stop. So what you're looking at there is, basically an open loop kind of emergence thing misses the whole point, that this thing is a homeostatic error reduction scheme in anatomical space. It's at a particular point in morphospace that describes a limb. If you try to deviate it, it will try to get back there. Another thing that you might not have heard of that's, I think, even more instructive is what happens when you take a tail and surgically graft it to the flank to the side of a newt or an axolotl. Over time, that tail remodels into a limb.
Oh, I didn't know that.
Yeah,
so you can put the wrong piece of anatomy on another part of the body, and it will turn into what it should be.
Correct. And if you look at, and the most instructive thing is to look at the tip of that tail, there's a bunch of cells there that turn into fingers. Now, these are tail tip cells sitting at the end of a tail. There's nothing locally wrong. Why are they turning into fingers? Because just like in cognitive systems, there's an element of top-down causation that what happens is, in the service of a large-scale goal, no individual cell knows what a finger is or how many fingers you're supposed to have, but the collective absolutely knows. And how you know it knows, this is not philosophy or linguistics, the reason that you know it knows is because you do the experiment, and if you don't understand that this thing is going to reduce error from this large-scale pattern, you're going to make the wrong prediction. The reality is, it's going to try to get back to the correct state, either by growing something new or remodeling something in place. There is a very strong influence of this set point on the lower level subunits. So all of the cell behaviors, all of the molecular, the gene transcriptions and the methylations and God knows what else has to happen in order to turn the tip of a tail into some fingers. All of that gets triggered not by any kind of local error. There's nothing wrong with those cells, by this large scale pattern minimization scheme to a specific goal. Okay, and so this is what I mean. This is very much an experimentally based cybernetic kind of model. So we've looked, and so now so now your question, okay, well, how the heck does it know what it's supposed to grow? And so I'm going to give you there's a few examples we can talk about.
Where is the set point encoded, and how is it?
Yeah, exactly. Where's the set point encoded? Because and it's really important because when you change the thermostat setting at your house, it's very critical that you don't need to know how the entire heating and cooling plant works. You're not going to micromanage that thing. All you need to know is A, that it is a homeostat, and B, how to change the set point. And after that, everything goes well. The reason that's important is because in regenerative medicine, if we want to be able to regrow limbs, to normalize tumors, to repair birth defects, we could try micromanaging it from the bottom up, which is what molecular medicine is trying to do today. I think that's a losing proposition in many ways. But also, we can take advantage of the native intelligence of the material and find the trigger points and the set points that we can rewrite and let the thing do what it does best, which is to execute to those set points. So this is the basis of our regenerative medicine approach in our lab. So I'll give you one example that's kind of one of my favorites. So imagine a flatworm. So this is a planarian. It's got a head and a tail, central nervous system, true centralized brain, 2 eyes in the head, and a very particular head shape. If you cut that thing into pieces, what you'll find is that every piece regrows a perfect little worm. Okay, so every piece has the information needed to remake an entire worm. So they're highly regenerative. You can ask the question, how does it know how many heads it's supposed to have? It's an interesting question because just saying, well, it's in the DNA, that doesn't help. We know the DNA doesn't contain things like head number or head shape directly. The DNA codes for proteins. So if you look at a wound, when you cut a planarian in half, just imagine, head here, tail here, you cut it in half like this. The cells on this side of the cut need to make a tail. The cells on that side of the cut need to make a head. They were near neighbors before you cut them apart. Why do they have different anatomical fates? It's not a local decision. You need to know what's the rest of the fragment doing, right? So we started studying this, and what we found so this compresses like 12 years of work. Long story short is that we found that there is a bioelectrical pattern across the entire worm that we partially decoded that says how many heads you're supposed to have. And what we found is that if we go in and rewrite that pattern, and the way that we rewrite that pattern, in the case of the planarian, we use drugs that open and close certain ion channels. In the case of the frog, we've used optogenetics. And when you say
that pattern, just to be concrete here, do you just mean there's a polarity, like a single gradient that goes from one end of the tissue to the other? What is the pattern?
No, it's more complex. It's more complex than that. I mean, in the planarian, it's like actually almost that simple. But in many of these cases, it's a much richer pattern. So and in fact, it's often quite complex. And we're working on software to try to deconvolve it the way that, when you do neural decoding, somebody's looking at a picture of a rose, you don't look in their brain and see a rose, right? You see a bunch of crazy signals that you have to somehow interpret. It's a little like that. There are complex spatial patterns that you can see, and you have to try to figure out which of those, what aspects of that pattern encode what organ shape, orientation, size.
But you're talking about a time evolving voltage pattern across all the cells.
Correct. And when I say time evolving, yes, it can evolve with time, especially in development, but overall it's quite, especially in adults, planaria or adults, it's quite stable. So you could look and it just sits there, and then this is, it's quite a rich, I mean, if you go to my website, you can see videos of and images of all this stuff. So what we figured out how to do was to reset that pattern so that by opening and closing certain ion channels, so that instead of being the one head pattern, it becomes a 2-head pattern.
You can induce the tissue to regenerate 2 heads when it should have done 1, and you do that by disturbing voltage patterns by playing with ion channels and which ones are open and closed and things like that.
Yeah, but the key here is that we're not just disturbing them. This is not sort of whack the television set until it does something. This is a very specific. We have a computational platform that, given a particular circuit with known ion channels and whatnot, it can help you derive, if you want the pattern to be this instead of this, which ion channel should you open and close. That's really a non-trivial problem. But for example, this is how we do repair of birth defects, because oftentimes there'll be a birth defect where you can see the pattern is incorrect. defect, and we know that if that goes on, there's going to be a very severe craniofacial hard gut defect. We can actually go in by using that computational platform and say, okay, I'm going to fix that pattern and to be the normal pattern. Then the model will say, well, to do that, you need to close this channel and open that channel and tweak some gap junctions or whatever it is. And if you do that, then the pattern changes shape, and then you get the repair. So in the case of it's very targeted stuff. In the case of the Planarian, yeah, it turns out that if you change that voltage, you get two-headed worms. And the amazing thing about it, so here are some amazing things about it. First of all, it's extremely modular in the sense that I didn't have to tell it what genes to turn on or what cells belong in a head or how to make a head. We don't have any idea how to do any of that. All we said is at the very highest level, you should have two heads. And that propagates down to the cells and the molecules, just like when you and I are talking. I'm not worried about tweaking the synaptic proteins in your brain to make you remember what I'm saying. You'll do that yourself. I'm giving you a very high level signal. And with our cognitive systems, just like that, are multi-scale kind of organizational architecture. So incredibly modular. We didn't have to sweat the details. Even things like size control, we didn't have to make sure that the head was matched the right size. It already does that. By the way, if you try to cheat and you do this molecularly with, for example, Wnt pathway modulation, as other people have done in the field traditionally, you can get extra heads that way, but those heads are not scaled correctly. Because you turn them into a head, but you didn't handle the size control. The bioelectric switch is way on top of that. Once the thing's committed to a second head, it will handle all the stuff.
I see this. So in those cases, molecular biologists were playing with one signaling pathway that was important for headness, but they couldn't tune everything that needed to be tuned because they were operating at sort of a lower level of information.
Exactly. And I'm not saying they couldn't. I mean, eventually, if you wanted to be in charge of 100
if you knew what all the other knobs were that you needed to do in parallel, but you're saying you can go above that.
You can go much higher, right? Yeah. The question is, to me, with hopes of regenerative medicine, the question is, what's the highest I can go? I want the least amount of effort. I want to know the least I have to know to micromanage to get the effect I want. So the other amazing thing about it is that if you take those two-headed worms and you chop them into pieces, guess what you have? You have more two-headed worms. In other words, once that bioelectric medium that stores that information has a memory to it. So once you've shifted it from a one-headed form to a two-headed form, it holds. And it holds, as far as we can tell, forever. It's been now several decades that we keep cutting these things. And so that's really important, because genetically, if you were to sequence those worms, you would be none the wiser that their body plan is completely different. In other words, this really is information, much like, again, to a molecular genetics person, this is crazy, because you've made a new lineage of permanent animals that is not at all reflected in the genome. But to a neuroscientist, this makes perfect sense. We remember things all the time. We don't have to change our genome to store new patterns. So this medium is able to store and hold permanent memories, and then we know how to turn it back as well.
Why? We have limited time, so I'm just going to give you some high level questions. Why don't all organisms have regenerative capabilities like planarians? That would seem at first blush to be like what you'd want to do. Like, yeah, you can just chop me into pieces and then it'll just create more of me. What are some of the fundamental trade-offs what are we trading off when we get rid of these kind of regenerative abilities in an organism like us that can't, you can't chop me in half and create two new nicks?
A few things. First of all, I like to tell people that, planaria are amazing and that you can regenerate a whole animal from a small fragment, but half the human population can regenerate an entire human body from one cell. So I actually disagree that we've lost it completely. What we don't tend to do is have it in adulthood. Some mammals do. So deer regenerate their antlers every year, like, 1.5 centimeters of new bone per day that they make. Right.
So the species level, you're saying we haven't lost regenerative capabilities, period. But as we develop into adults, like at the individual level, we don't have the regenerative capacity that an individual planarian has. But at some point in our development, that capacity goes away or goes down.
That's correct. And at the beginning, even the individuals have it. So if I take an early embryo and chop it into pieces, I don't get half bodies. I get twins and triplets and things like that, right? So at the beginning, we all still have it. Yeah. Okay, I'm going to tell you a sort of a kind of a superficial story, and then a deeper story for why this is. The superficial story goes like this: Look, you're an early mammal. You're running around the forest. Somebody bites your leg off. What's going to happen? Well, unlike an amphibian, first of all, you're going to try to put weight on it. So whatever delicate blastema you're trying to grow at the end of that wound is going to get rubbed into the forest floor. That's the first thing. The second thing is that you're probably going to get infected. You have a high blood pressure. You have a rapid heartbeat. You're going to lose a lot of blood. You're probably going to get infected from walking around. You're never going to live to regenerate anything. Just scar, seal the wound, lots of inflammation, seal the wound, and hope to live another day. It's interesting that the one mammal that does regenerate does it in a non-load bearing appendage. It's because the antlers are hanging around up there and nobody's trying to put weight on them. There's something else which is interesting, which is that almost all the good regenerators are aquatic. And that's because in dry air, it's very hard to drive the ion currents out of your wound epithelium that is required for regeneration, which is why in our efforts, and we're working on regeneration in mammals now. We solved it in frog. We're working on it in mice. We provide a wearable bioreactor that provides an aqueous almost amniotic environment for these things to happen. So the idea was it was a trade-off because it wasn't worth it the way it is for amphibians to try and put energy into that. The deeper story, but I'll tell you what I think is a deeper story about what's going on in Planarian. When most animals reproduce, their offspring don't inherit their somatic mutations. The bottleneck of sexual reproduction cleans off whatever happened to your soma during your lifetime, right? They don't get those changes. You start kind of fresh each time. Planaria are not like that. At least the planaria that we study are asexual. The way they reproduce is they rip themselves in half and then they regenerate. So what happens then is that any mutation that doesn't kill the cell that it's in, the neoblast or the stem cell that it's in, participates in creating part of the body, and so it makes it into the next lineage. So planarian genomes are incredibly messy. They're mixoploid. Every cell might have a different number of chromosomes. They're like a total mess because they keep all that stuff. And so what planaria are dealing with is a very unreliable substrate. Now, all biological substrate is unreliable because unlike with our computer technology, in biology, everything's going to decay at some point. You never know how many copies of anything you really have. It's all sort of very it really is kind of unreliable. But in planaria, it's incredibly unreliable. And what you find, and we've done simulations to show this, is if you do evolution on an agential material that is simultaneously unreliable but also quite competent. In other words, the cell groups can learn things, they can make decisions, they can try to reduce errors and so on. When you evolve over that material, the winning strategy, and if you want, I can go into it. But what actually happens is that evolution spends all of its time cranking on the algorithm, meaning let's keep our genome as clean as we can, but let's make the best we can, knowing that the hardware is kind of junky. What algorithm is going to let us get to what we need to do? And so what's incredible about planaria? Here's some interesting facts about planaria. Every other model system you can call a stock center. You can get fruit flies with the funky wings and mice with curly tails, and you can get lines of mutants. There are no lines of mutants of planaria except for the two-headed form that we made, and those are not genetic, right? Also, there are no transgenic planaria. Nobody has shown a transgenic planaria because this is kind of a funny way of putting it. They just don't take their genetics all that seriously. They assume from the beginning that the genome's going to have all kinds of junk in it. And what's really what all the effort has gone into is working out the mechanisms, including bioelectrics, no doubt other things that allow you to do this, despite the fact that there's going to be your hardware is error prone. So I think there's a continuum, right? C. elegans is on this end. As far as we know, they basically just do one thing, and that's what they do. Planaria are all the way. This went all the way, and then amphibians and mammals are sort of in between. Like we can make up for some things, but not too many things. So I think what's happening here is a set of trade-offs in planaria forced by the fact that there really is there is no good way to do quality control on the hardware, but for mammals and even amphibians there kind of is. So there's a bit of a trade-off. Here's the craziest thing. This is what I started off with for students. Whoever told you in any of your biology classes anything that would lead you to expect that the animal with the messiest genome would be the one that was cancer resistant, highly regenerative, does not age, immortal. They don't age, right? And it's not the animal with the cleanest genome; it's the animal with the messiest genome. Amazing, right? Like this is exactly the opposite of what we usually are led to believe. I think that's what's happening.
Yeah, and so I want to get back to this question of, what if we have an end in mind, like the creation of a limb or an organ or whatever, where is that set point encoded, and how is it encoded if it's not in a place like the genome? Because normally when we start to ask questions like this, I can remember when I was younger, right? People, literally myself, we used to think, oh, it's all in the genome. Everything's pre-specified in the genome, and of course it's not. So where does the end product live that you're constantly using, these error correction mechanisms to move toward?
Yeah. Okay. Again, I'll tell you the kind of more or less the more or less solid thing, and then some speculation above that. The solid thing is this, that you can't say it lives in the DNA either, actually, because
Right.
we can read genomes now. We know what's there, and what's there is information about proteins and some timing information about when they're going to show up in different contexts. There is nothing that you can read out of a genome directly that says, oh yeah, two eyes, bilateral symmetry. Well, except for the heart, that one goes on the left. Well, what's the left? You can't encode that genetically. So none of this is in the genome, and we're going to have to, as I'm going to point out in a minute, we're going to have to redefine the whole question of what anything being in anything is anyway. But from that perspective, the genome is no closer than anything else in telling you where things live. What the genome creates is some hardware. Okay, so some biochemical hardware, and some of that biochemistry actually also has biomechanical and bioelectrical properties, and we study the bioelectrical ones. And that hardware has the genomically specified hardware has some interesting properties. Imagine you buy a calculator at the store. You turn it on. The very first time you turn it on, it very reliably, it says 0. It always says 0. Now, you might be forgiven if you've never heard of a reprogrammable calculator. You might be forgiven to say, oh, I've been given some hardware that encodes 0. And in fact, here's the schematic of the calculator, and here are where all the transistors are. And now I can say all of those things encode the pattern that goes like this, right? And that's fine. But if you know that it's reprogrammable, you find out that, besides the default, very reliable default state, you can also put other things in there. And what you've actually been given is something much more interesting. You've been given some hardware that, by default, it has a particular pattern that it creates anatomically, transcriptionally, physiologically, and so on. But it also has the capacity to store other patterns that can be rewritten by experience. So this should sound completely familiar to anybody who does neuroscience and cognitive science. And so if you want to ask where are the patterns stored, that we know the answer to, because I could show them to you, and I can also show you how to rewrite them. So they are stored in a spatiotemporal pattern of electrical states in the tissue, just like presumably the question of where is our cognitive processes stored. They are stored in a much slowed down version of the exact same thing that happens in brains, which is slowly changing electrical patterns, moving through the network, doing computations, trying to figure out what the symmetry is, who's going to be head, who's going to be tail, how many eyes are we supposed to have, exactly the same stuff. You can see them, we can rewrite them, and so that answer has been, that question has been answered. There's a deeper question we can ask, which is, in general, the question is, where do patterns come from in the first place? Like where do these patterns actually originate? And so here's the more speculative thing. And if you're interested, I'm currently running this whole asynchronous symposium. It's called the Symposium on Platonic Space, and it's all fall. People are doing talks on this. It's very interesting. We as biologists, when we talk about where do patterns come from, we tend to love two kinds of answers, and most things are a combination, we think, of these two answers. One kind of answer is constraints of physics, meaning that, well, it's trying to minimize surface area, so it's got to be round, and adhesion means this or that, so it's physics. And the other thing we love is evolutionary history. It looks like this because everything else got killed off, and this is what you have left, right? So there's some kind of history of interactions with the environment that makes sure that you look exactly like this and not like that. So, and that's fine for things that have an evolutionary history. Even then, both in physics and in biology, there's something we already know those two things aren't quite right, because imagine, and this is just an example, and now we've really sort of cranked the knob on this one. Imagine cicadas. The cicadas come out at 13 years and 17 years. And so as a biologist, you want to understand why that is. You say, so why is that? And well, it's because it's so that their predators don't time their cycles, right? If it was 12, then every 2-year predator, every 3-year predator, every 4-year predator. So you say, oh, okay, amazing. And so these are prime numbers then. Sure. Why 13 and 17 specifically? And now you need to go understand the distribution of primes. And now you're no longer in biology, you're in the math department. And because the answer to that, so as you said, like every kid keeps asking why. So with all of these questions, physics too, if you start asking questions about, say, so why do fermions do this or that? It's because this mathematical object has a certain symmetry and there's two kinds, and all of these things eventually end up in the math department. Mm-hmm. If you really want insight into what's going on, you end up in explanations that are not biology, they're not physics, they're properties of mathematical patterns. And if the mathematical patterns were different, the physics would be different, but also the biology would be different. In other words, cicadas would be coming out at different years if the prime numbers were differently organized. So we have to start thinking that some of the reasons for why certain patterns are the way they are not evolution reasons, and they're not physics reasons, they're actually math reasons. And so here's where the rubber really hits the road. We have been making creatures that have never existed before. So these are, as have other people, but we've made Xenobots, Anthrobots, large-scale systems that have never had a history of selection for this embodiment. The cells come from other creatures, but there's never been selection to be a good Xenobot or a good Anthrobot. If you want, I can tell you what some of them do. But the bottom line is they have very specific and very unique transcriptomes of physiological states, behaviors, shapes, these things, all of these things, very specific, very new.
Let's be concrete for people. What are you literally talking about right now? Like when you say Xenobot, what have you done in the lab?
Okay, so you isolate some cells from the epithelia of a frog embryo, okay, and you set them aside. You don't touch the genome, so no synthetic circuits. No,
you're taking cells from an existing creature.
You're taking cells from an existing creature. No scaffolds, no weird nanomaterials. You set them aside, and you let them reboot their multicellularity. They could do many things. They could crawl away from each other. They could die. They could form a 2-dimensional monolayer like cell culture. Instead, what they do is they come together into a little spherical looking thing. They have cilia on the outside. They coordinate the beating of those cilia so that the thing can start moving through water. They have various behaviors, and then they can do all kinds of interesting things. For example, they have over 600 differentially expressed genes compared to what they were doing back when they were inside the embryo. And remember, the only thing we've done them is free them from the bullying by other cells that make them to be like this boring 2-dimensional cell, skin surface. Taking them away from those other cells reveals that by themselves turn on hundreds of new genes. Some of those genes are responsible for hearing and vibration, sound processing. And in fact, we found out that, yes, unlike embryos, Xenobots do respond to sound that we provide with a speaker under the dish. They also do this crazy thing called kinematic self-replication. So if you sprinkle a bunch of loose epithelial cells in their environment, they run around, they collect those cells into little balls. Those little balls mature to be the next generation of Xenobots. And guess what they do? They run around doing the same thing. So you have generation after generation after generation.
Basically, you isolate some cells, some boring cells from an existing creature, and they will essentially reprogram themselves and behave like a new type of creature.
Yes, to be very clear, I'm not saying the cells were boring. I'm saying the other cells
I know.
were confining them to a boring fate.
Yeah. They had all of this potential inside of them.
Yeah. And the human version of this, so let me just tell you about the human version of this, because it's completely wild. At that, when we made Xenobots and we were saying, like, look, there's never been selection to do kinematic self-replication or to have these genes. Like, there's never been any Xenobots. Where's this coming from? And people said, well, they're amphibian and they're embryonic, so maybe there's plasticity. I said, fine. What's the furthest you can get from an embryonic frog? Well, how about an adult human? So what we did was we took tracheal epithelial cells donated by adult human donors for biopsies. Same thing, no genetic changes, no scaffolds, no nanomaterials. We let them self-assemble. They self-assemble into a little sphere. They turn themselves inside out so that the cilia, again, are on the outside. They start swimming around. 9,000 new differentially expressed genes over what they would have been doing. Okay, so half the genome. They completely reorganize. So their environment's not that different. They're still.
And what is the environment? Like, what are you putting them in?
Originally Matrigel, and then just liquid media. I mean, it's designed to be as simple to the milieu of
simple aqueous environment, basically.
There's nothing else. Right. People say, well, it's environmental. Where's the specificity? The environment doesn't tell you to any of those things. The environment is just like, it's like a normal cell culture medium. There's nothing in there. So they can do some interesting things too. They have 4 distinct behaviors that they can do in terms of moving around. They have, like I said, 9,000 differentially expressed transcripts. They have the ability, if you take a petri dish, plate a bunch of iPS cells that turn into neurons. So you make a iPS derived human neuron culture, and then you take a scalpel and you put a big scratch through it. Okay, so make a big wound. Throw some anthrobots in there. The anthrobots sort of running around. They find the wound. They drive down the wound. They pick a spot and they sit. And when they sit, 4 days later, you say, what are they doing? You lift them up, and what you see is that what they were doing is taking the 2 sides of the wound and healing it together. We had some amazing pictures in this in these papers that we put out, where you can see that what they were doing is healing the 2 sides of the neural wound. So they also have who would have thought that your tracheal epithelium that sits there for decades dealing with mucus and air particles has the ability to self-organize into a self-motile little creature that will radically transform its transcriptome, that will have the ability to heal other kinds of tissues. Who knows what else it can do? They're younger, by the way. If you look at epigenetic age, they're younger than the cells they come from. So they also roll back their aging clock. Like, there's never been selection for any of this. They don't look like any stage of human development. This has never existed before. And so we really need to understand, where do these things come from? Right? Where do the very specific endpoints? Why 4 behaviors? Why not 9? Why these genes, not some other genes?
So, and right? I guess what you're saying is, when you look at these I see what anthrobots means now. You literally take human cells and they turn into these little things. When you take these things and you observe them, they do all of this interesting stuff. They reconfigure their genomes. They turn into these little anthrobots that do all these things you would never expect a clump from someone's trachea to do. You're saying all of the stuff that they're doing can't be encoded in the environment, because the environment's this very simple medium that doesn't have the information in it that it would need. And it can't be, you can't resort to an evolutionary explanation, because there's no history here that would tell you why this type of cell would behave this way in this new environment. Like, there's no history that's selected for that. So what's left in terms of the possible explanations?
Yeah, and I just want to first mention, because people will jump on this point. They'll say, oh no, the human had a history, and at the same time that the genome learned how to be a human, it also learned how to make anthrobots, right? So the problem with that explanation, so I just want to kind of address it up front, because I'm sure people listening to this will some of them are thinking this. I'm going to address it up front. The problem with that explanation is that the whole point of the theory of evolution was supposed to be able to explain great specificity of why a certain being is the way it is, with the specific history of the forces that acted on it. That's the whole point. Yeah. If you're now telling me that, oh yeah, and also a bunch of other stuff that was never part of the history and that you couldn't possibly have seen coming, that kind of undoes a good chunk of the point. Like, there's supposed to be this specificity. You can't just say, yeah, well, okay, and many people do. They say, oh, it's emergent, and what does that mean? Well, it's just something that holds in this world. I find that completely unsatisfying. I think if we're going to have theories, they need to tell you what is the space of possibilities from which this was picked, and why this specific thing other than something else. There's also a computer science question here, which is that computations cost energy and they take time, which is, we know when the human was computed in the eons of bashing the genome against certain environments. That's when you computed how to make a human. When did we pay the computational cost for Xenobots or Anthropots? We never did. There was never that process never took place. So there's something really missing with the standard account, I think. And so now the key question: well, what else could possibly be left? Okay, so there's 2 approaches to this. One approach is to say these things are just emergent. There's lots of emergent surprises. And I find this completely unsatisfying, because I don't want to have a big book of emergent surprises that we write them down in, and then sort of that's that.
But that seems kind of close to just saying, well, it's a miracle.
Basically, and it's funny because what I'm about to say, a lot of people would say that's the more sort of woo-woo explanation, but I actually think it's the other way around. The standard explanation is very Mysterian. It's very Mysterian. It's very pessimistic. It just means that we're going to be surprised from time to time. We're going to record all these things, and they're just things that hold in our world. I find that totally unsatisfying. I prefer a more optimistic idea, which is this. Let's suppose, and there's a precedent for this. The precedent is mathematicians. So there's a lot. I don't know if probably most. I haven't done the survey, but many, if not most, mathematicians think that they are not creating, think they are exploring and discovering a pre-existing set of truths. There is a space of mathematical facts, things like the actual value of E and Feigenbaum's constant, and facts about why you can have formulas to solve equations of degree 4 but not 7. It's impossible. Like why? Like all these things, right? There are all these facts about mathematical objects that do not have an explanation in terms of selection or physics, and they think they're exploring those. We already know that a lot of those patterns are important for both physics and biology. I think biology exploits those patterns like crazy because they're amazing free lunches. They save evolution time. For example, let's just imagine that there's some world in which selection, the highest fitness in this world belongs to a triangle of a very particular shape. So evolution cranks a bunch of generations and it finds the first angle, great. And it cranks a bunch of generations and finds the second angle, great. Now you save 1/3 of the time. You don't need to look for the third angle. You know exactly what it is because there's this magical fact of geometry that tells you what it is. If you got the first two, you know what the third angle is. You don't need to evolve it. And there's tons of stuff like this where if you just do certain things, like if you evolve a voltage-gated ion conductance, like a voltage-sensitive ion channel, you got a transistor. With a couple of those, you can make logic gates. You get the whole truth table and all the cool properties of logic gates. You don't have to evolve the truth table. You get it for free from mathematics. That's just how it is. You don't need to spend the time doing that. So my point is this, that what I think, and I go into this in great detail in one of the talks on this symposium page, that what I think is happening here is that there is a structured, ordered set of patterns. Many of these patterns are very simple things that mathematicians study, but others of these patterns are quite complex, dynamic patterns that are facilitated by physical interfaces. What are those physical interfaces? They're anything from what physics recognizes as particles to the things that biology studies, which is if you set up certain materials in a certain way, you get the benefit of a whole set of patterns that are there for you because they are facts of mathematics. Some of these are very simple things about the geometric shapes or whatever, or computational things, or very complex things like shapes of behavior. So some of these things actually, I think this is all relevant to neuroscience in that I think these patterns don't just set the structure of the body. I think to a large extent, they set the structure of cognition. I think some of these things we would recognize as behavioral propensities. So that's what I think. I think that and then there's a research agenda of systematically mapping out that space by creating novel interfaces. So the normal embryo shows us one point in that space. If you just let the frog genome do what it does, you get, we know what you get. You get this like one thing. But what's the stuff around it? What are the other things it can do and why? And so by making various chimeras and Xenobots and anthrobots and all this stuff, we can basically explore that latent space of possibilities and get to an understanding of the mapping between the interface and the patterns you get to really understand from this latent space of possibilities, why do you get specific complex outcomes at specific locations in specific scenarios. So that's what I think is going on.
So was Plato right?
First of all, I mean, okay, the only reason I call it Platonic space is not because I want to stick close to what Plato said, to whatever extent we know what he said. I use it, and I may at some point need to change the name, but for now, I use it because the mathematicians know exactly what I'm talking about. Mm-hmm. Because it links, because this goes from, I mean, when let's face it, when biologists first hear this idea, they think this is nuts, because we have been sort of conditioned in our education to like a very sparse ontology. Nobody wants to be thinking about another realm of patterns.
Right.
We've got some chemistry, and we've got this three-dimensional space in which chemicals move. That's what we prefer. And in fact, most people would rather take the mysteries of emergence over thinking that there's this whole other space around.
Well, and I noticed too, it was interesting to me as you were saying all this, you said your preference is not for that type of explanation. It's for the one that you just described to us. But you said it was your preference, and other people have a preference for a different type of ontology. How much of this boils down to just almost these aesthetic preferences that people have or don't have?
I think a lot of it, a lot, and these are, I would call them metaphysical preferences, because I can't prove. There's no rigorous proof that I can give you that that emergence is the wrong way to think about it, but in the sort of these metaphysical preferences, like would you rather have a bigger ontology where you accept the existence of these other things, or would you rather lose predictive power somewhere else? But what I think is very important is this: while it starts with metaphysical preferences, that's not where it ends, because where it ends is in the clinic. In other words, I'm not saying that this is a philosophical view, and if you prefer it, you can take it. If you prefer, you take the other view, and it doesn't matter, and we'll never know who's right. I don't think that's correct at all.
You're saying we either will or will not derive concrete outcomes that can be applied in the clinic from doing things, the version A versus version B.
Exactly. Exactly. I think these views drive very different research programs. They make certain things invisible or inaccessible. I mean, all models shut off something to enable something else. But I think the only value in all of this stuff is that if it drives research, and I would claim that it already has in our group and other places, where we can already see that by following some of these things, we've discovered new science that nobody has discovered before. Mm-hmm. The thing with just to say the thing with the planaria, the first two-headed worms were found in 1903 or 1905 or something like that. In 100 years between that and 2008, when we were the first to do it, nobody recut them. To my knowledge, nobody ever recut them to see what would happen, because it was completely obvious. Because, well, the genetics say one head, why, then of course it'll go back to normal if you cut them. Yeah, but it was not, you couldn't, with the standard view, you could not see this experiment with the standard view, right? And there's tons of stuff like that. So yes, it starts as metaphysical preferences, but it ends, the rubber hits the road at the bench. Either these things will or they will not lead to new science. And so, I think also it's very important that we understand it isn't about explaining after the fact, because you see, you can always tell a molecular story if you're so inclined. So somebody sees the two-headed planarian and go, oh, well, yeah, I turned on some genes to make a head and with eyes. And I mean, yeah, that has a molecular explanation. Right. I'm not saying it's magic. When you look underneath a hood, you're never going to see fairies. It's always going to be chemistry. But there's a difference between after somebody's done something new saying, yeah, I suppose that's consistent with the laws of physics. Yeah, great. That's not what we're looking for. We're looking for frameworks that would help you find the new thing that you otherwise would never have found. And so I think it's really it's really important that we going back to this name thing. The reason I start talking about this as Platonic Space is I want to be clear with people. This isn't new. This isn't some new crazy thing that I invented. This has been with us for the longest time. Plato and Pythagoras especially have been on top of this and some Eastern thinkers, of course, for a very long time. The mathematicians already knew this. In Newton's boring classical universe, that was already haunted by the non-physical laws of mathematics, that certain physics is the way it is because of mathematical truth. So, we already knew all this. And so now the only question was evolution blind to this? Has evolution somehow failed to notice that you can exploit these amazing free gifts that you don't need to, that are very useful, but you don't need to evolve them? I don't believe for a second that evolution was failed to notice this. It uses this all over the place. And so was Plato right? I think he was absolutely right about the importance of a non-physical space of truths that impacts biology. I think he was 100% right about that. As far as I can tell, he really thought that those patterns or those forms are unchanging and eternal. They're kind of fixed. And that one, I mean, who am I to disagree with Plato? But on that, I don't believe that. I actually think that space is not static and unchanging. I think that in some way that we're still working out a good, quantitative model for. They change via their interface in the physical world. I think every time they impact the physical world, I think there's bidirectional feedback of some sort. And B, I think that, and this is something that we're actually testing in our lab now, so stay tuned. We'll see what we find. I actually think there's some compute that gets done in that space. I don't think all the compute gets done in our space here. I think that everything that we know about the computer science, the Turing model and algorithms and all of that. I think that's a model of the front end interface. It's not a model of the whole thing. It's all of these things are models of like of the physical interface to these patterns. But I think there's both information that comes from that space, and I think there's some compute that that can be done in that space that we might be able to exploit. And that would have, if that's true, that would have very practical implications for computer science and so on, and all of that will be visible. Like that will all be empirical results, not philosophy.
Is there anything you want to reiterate, or any final thoughts you want to leave people with on the space of subjects we covered in this 1 hour podcast?
I'll just end in one thing, that given that we already know that there are some reasons why the kinds of processes we see in neuroscience are an amazing interface for the things that we observe in behavior. So various cognitive competencies. And so now we have a stepping stone, which is to consider how those things play out in other body tissues, because what I'm pointing out is that there is no rigorous reason to say that your liver, albeit at a much slower rate, is doing many of the things that you see your brain doing. Just because it doesn't talk, it doesn't have language, it doesn't mean it's not doing some of those things. This is really a good stepping stone in an emerging field called diverse intelligence to ask what other kinds of systems are potentially interfaces for radically different minds that we are very bad at noticing. And so there are some new tools emerging in neuroscience, such as causal information theory, right? Like the kinds of stuff that Tononi and Eric Hole do and so on, that allows us to kind of try to say, so what is happening in the mind, in the brain, that allows a mind that's more than the sum of its parts? Where else might that appear? And I'll just reiterate my very strange claim at the beginning, is that I don't think neuroscience is about neurons at all. I think neuroscience is a science of the scaling of minds. And there's a very interesting field called diverse intelligence, which not only asks where did our cognitive capacities come from in evolution, but actually what's the substrate dependence here? If it doesn't have to be nerves, does it even have to be cells? Like what's the actual... Right? What's the actual secret sauce here? And what other kinds? And so there's a whole other talk that I give where I talk about very diverse kinds of minds. And so I think, neuroscience, I think is critically important for a ton of different fields, including physics, including, environmental science and whatever, but not because of the nerves. It's not because of the neurons. It's because of what the neurons are fundamentally trying to tell us about the scaling of minds in this world.
All right. Well, Dr. Michael Levin, thank you very much for your time.
Thank you so much. Yeah. Thank you for that.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.